Welcome to the I Can't Sleep podcast, where I read random articles from across the web to bore you to sleep with my soothing voice. I'm your host, Benjamin Boster. Today's episode is from a Wikipedia article titled Neuron. Within a nervous system, a neuron, or nerve cell, is an electrically excitable cell that fires electric signals called action potentials across a neural network. Neurons communicate with other cells via synapses, which are specialized connections that commonly use minute amounts of chemical neurotransmitters to pass the electric signal from the presynaptic neuron to the target cell through the synaptic gap. Neurons are the main components of nervous tissue in all animals except sponges and placozoa. Non-animals like plants and fungi do not have nerve cells. Molecular evidence suggests that the ability to generate electric signals first appeared in evolution some 700 to 800 million years ago, during the Tonian period. Predecessors of neurons were the peptidergic secretory cells. They eventually gained new gene modules, which enabled cells to create postsynaptic scaffolds and ion channels that generate fast electrical signals. The ability to generate electric signals was a key innovation in the evolution of the nervous system. Neurons are typically classified into three types based on their function. Sensory neurons respond to stimuli such as touch, sound, or light that affect the cells of the sensory organs, and they send signals to the spinal cord or brain. Motor neurons receive signals from the brain and spinal cord to control everything from muscle contractions to glandular output. Interneurons connect neurons to other neurons within the same region of the brain or spinal cord. When multiple neurons are functionally connected together, they form what is called a neural circuit. Neurons are special cells which are made up of some structures that are common to all other eukaryotic cells, such as the cell body, soma, a nucleus, smooth and rough endoplasmic reticulum, Golgi apparatus, mitochondria, and other cellular components. Additionally, neurons have other unique structures such as dendrites and a single axon. The soma is a compact structure, and the axon and dendrites are filaments extruding from the soma. Dendrites typically branch profusely and extend a few hundred micrometers from the soma. The axon leaves the soma at a swelling called the axon hillock and travels for as far as 1 meter in humans or more in other species. It branches, but usually maintains a constant diameter. At the farthest tip of the axon's branches are axon terminals, where the neuron can transmit a signal across the synapse to another cell. Neurons may lack dendrites or have no axon. The term neurite is used to describe either a dendrite or an axon, particularly when the cell is undifferentiated. Most neurons receive signals via the dendrites and soma and send out signals down the axon. At the majority of synapses, signals cross from the axon of one neuron to a dendrite of another. However, synapses can connect an axon to another axon or a dendrite to another dendrite. The signaling process is partly electrical and partly chemical. Neurons are electrically excitable due to maintenance of voltage gradients across their membranes. If the voltage changes by a large enough amount over a short interval, the neuron generates an all-or-nothing electrochemical pulse called an action potential. This potential travels rapidly along the axon and activates synaptic connections as it reaches them. Synaptic signals may be excitatory or inhibitory, increasing or reducing the net voltage that reaches the soma. In most cases, neurons are generated by neural stem cells during brain development and childhood. Neurogenesis largely ceases during adulthood in most areas of the brain. Neurons are the primary components of the nervous system. along with the glial cells that give them structural and metabolic support. The nervous system is made up of the central nervous system, which includes the brain and spinal cord, and the peripheral nervous system, which includes the autonomic, enteric, and somatic nervous systems. In vertebrates, the majority of neurons belong to the central nervous system, but some reside in peripheral ganglia, and many sensory neurons are situated in sensory organs, such as the retina and cochlea. Axons may bundle into fascicles that make up the nerves in the peripheral nervous system, like strands of wire make up cables. Bundles of axons in the central nervous system are called tracts. Neurons are highly specialized for the processing and transmission of cellular signals. Given their diversity of functions performed in different parts of the nervous system, there is a wide variety in their shape, size, and electrochemical properties. For instance, the soma of a neuron can vary from 4 to 100 micrometers in diameter. The soma is the body of the neuron, as it contains the nucleus. Most protein synthesis occurs here. The nucleus can range from 3 to 18 micrometers in diameter. The dendrites of a neuron are cellular extensions with many branches. This overall shape and structure are referred to metaphorically as a dendritic tree. This is where the majority of input to the neuron occurs via the dendritic spine. The axon is a finer cable-like projection that can extend tens, hundreds, or even tens of thousands of times the diameter of the soma in length. The axon primarily carries nerve signals away from the soma and carries some types of information back to it. Many neurons have only one axon, but this axon may, and usually will, undergo extensive branching, enabling communication with many target cells. The part of the axon where it emerges from the soma is called the axon hillock. Besides being an anatomical structure, the axon hillock also has the greatest density of voltage-dependent sodium channels. This makes it the most easily excited part of the neuron and the spike initiation zone for the axon. In electrophysiological terms, it has the most negative threshold potential. While the axon and axon hillock are generally involved in information outflow, this region can also receive input from other neurons. The axon terminal is found at the end of the axon farthest from the soma and contains synapses. Synaptic boutons are specialized structures where neurotransmitter chemicals are released to communicate with target neurons. In addition to synaptic boutons at the axon terminal, a neuron may have en passant boutons, which are located along the length of the axon. The accepted view of the neuron attributes dedicated functions to its various anatomical components. However, dendrites and axons often act in ways contrary to their so-called main function. Axons and dendrites in the central nervous system are typically only about 1 micrometer thick, while some in the peripheral nervous system are much thicker. The soma is usually about 10 to 25 micrometers in diameter, and often is not much larger than the cell nucleus it contains. The longest axon of a human motor neuron can be over a meter long, reaching from the base of the spine to the toes. Sensory neurons can have axons that run from the toes to the posterior column of the spinal cord, over 1.5 meters in adults. Giraffes have single axons several meters in length running along the entire length of their necks. Much of what is known about axonal function comes from studying the squid giant axon, an ideal experimental preparation because of its relatively immense size, 0.5 to 1 millimeter thick, several centimeters long. Fully differentiated neurons are permanently postmitotic. However, stem cells present in the adult brain may regulate functional neurons throughout the life of an organism. Astrocytes are star-shaped glial cells. They've been observed to turn into neurons by virtue of their stem cell-like characteristic of pluripotency. Like all animal cells, the cell body of every neuron is enclosed by a plasma membrane, a bilayer of lipid molecules with many types of protein structures embedded in it. A lipid bilayer is a powerful electrical insulator, but in neurons, many of the protein structures embedded in the membrane are electrically active. These include ion channels that permit electrically charged ions to flow across the membrane, and ion pumps that chemically transport ions from one side of the membrane to the other. Most ion channels are permeable only to specific types of ions. Some ion channels are voltage gated, meaning that they can be switched between open and closed states by altering the voltage difference across the membrane. Others are chemically gated, meaning that they can be switched between open and closed states by interactions with chemicals that diffuse through the extracellular fluid. The ion materials include sodium, potassium, chloride, and calcium. The interactions between ion channels and ion pumps produce a voltage difference across the membrane, typically a bit less than 1/10 of a volt at baseline. This voltage has two functions. First, it provides a power source for an assortment of voltage-dependent protein machinery that is embedded in the membrane. Second, it provides a basis for electrical signal transmission between different parts of the membrane. Numerous microscopic clumps called Nissl bodies or Nissl substance are seen when nerve cell bodies are stained with a basophilic, base-loving dye. These structures consist of rough endoplasmic reticulum and associated ribosomal RNA. Named after German psychiatrist and neuropathologist Franz Nissl, they are involved in protein synthesis, and their prominence can be explained by the fact that nerve cells are very metabolically active. Basophilic dyes such as aniline or weakly hematoxylin highlight negatively charged components, and so bind to the phosphate backbone of the ribosomal RNA. The cell body of a neuron is supported by a complex mesh of structural proteins called neurofilaments, which together with neurotubules, neuromicrotubules, are assembled into larger neurofibrils. Some neurons also contain pigment granules, such as neuromelanin, a brownish-black pigment that is a byproduct of synthesis of catecholamines, and lipofuscin, a yellow-brown pigment, both of which accumulate with age. Other structural proteins that are important for neurofunction are actin and the tubulin of microtubules. Class 3 beta-tubulin is found almost exclusively in neurons. Actin is predominantly found at the tips of axons and dendrites during neuronal development. There, the actin dynamics can be modulated via an interplay with microtubule. There are different internal structural characteristics between axons and dendrites. Typical axons almost never contain ribosomes, except some in the initial segment. Dendrites contain granular endoplasmic reticulum or ribosomes in diminishing amounts as the distance from the cell body increases. Neurons vary in shape and size and can be classified by their morphology and function. The anatomist Camillo Golgi grouped neurons into two types. Type 1 with long axons used to move signals over long distances, and type 2 with short axons, which can often be confused with dendrites. Type 1 cells can be further classified by the location of the soma. The basic morphology of type 1 neurons, represented by spinal motor neurons, consists of a cell body called the soma and a long, thin axon covered by a myelin sheath. The dendritic tree wraps around the cell body and receives signals from other neurons. The end of the axon has branching axon terminals that release neurotransmitters into a gap called the synaptic cleft between the terminals and the dendrites of the next neuron. Most neurons can be anatomically characterized as unipolar, Single process. Unipolar cells are exclusively sensory neurons. Their dendrites are receiving sensory information, sometimes directly from the stimulus itself. The cell bodies of unipolar neurons are always found in ganglia. Sensory reception is a peripheral function, so the cell body is in the periphery, though closer to the CNS than a ganglion. The axon projects from the dendrite endings past the cell body in a ganglion and into the central nervous system. Bipolar, one axon and one dendrite. They are found mainly in the olfactory epithelium and as part of the retina. Multipolar, one axon and two or more dendrites. Golgi 1, neurons with long projecting axonal processes. Examples are pyramidal cells, Purkinje cells, and anterior horn cells. Golgi 2, neurons whose axonal process projects locally. The best example is a granule cell. Anaxonic, where the axon cannot be distinguished from the dendrites. Pseudo-unipolar, one process which then serves as both an axon and a dendrite. Some unique neuronal types can be identified according to their location in the nervous system and distinct shape. Some examples are basket cells, interneurons that form a dense plexus of terminals around the soma of target cells, found in the cortex and cerebellum. Betz cells, large motor neurons. Lugaro cells, interneurons of the cerebellum. Medium spiny neurons, most neurons in the corpus striatum. Purkinje cells, huge neurons in the cerebellum, a type of Golgi 1 multipolar neuron. Pyramidal cells, neurons with triangular soma, a type of Golgi 1. Rosehip cells, unique human inhibitory neurons that interconnect with pyramidal cells. Renshaw cells, neurons with both ends linked to alpha motor neurons. Unipolar brush cells, interneurons with unique dendrite ending in a brush-like tuft. Granule cells, a type of Golgi 2 neuron. Anterior horn cells, motoneurons located in the spinal cord. Spindle cells, interneurons that connect widely separated areas of the brain. Direction. Afferent neurons convey information from tissues and organs into the central nervous system and are also called sensory neurons. Efferent neurons, motor neurons, transmit signals from the central nervous system to the effector cells. Interneurons connect neurons within specific regions of the central nervous system. Afferent and efferent also refer generally to neurons that respectively bring information to or send information from the brain. A neuron affects other neurons by releasing a neurotransmitter that binds to chemical receptors. The effect upon the postsynaptic neuron is determined by the type of receptor that is activated, not by the presynaptic neuron or by the neurotransmitter. A neurotransmitter can be thought of as a key, and a receptor as a lock. The same neurotransmitter can activate multiple types of receptors. Receptors can be classified broadly as excitatory, causing an increase in firing rate, inhibitory, causing a decrease in firing rate, or modulatory, causing long-lasting effects not directly related to firing rate. The two most common, 90% plus, neurotransmitters in the brain, glutamate and GABA, have largely consistent actions. Glutamate acts on several types of receptors and has effects that are excitatory at ionotropic receptors and a modulatory effect at metabotropic receptors. Similarly, GABA acts on several types of receptors, but all of them have inhibitory effects, in adult animals at least. Because of this consistency, it is common for neuroscientists to refer to cells that release glutamate as excitatory neurons, and cells that release GABA as inhibitory neurons. Some other types of neurons have consistent effects. For example, excitatory motor neurons in the spinal cord that release acetylcholine, and inhibitory spinal neurons that release glycine. The distinction between excitatory and inhibitory neurotransmitters is not absolute. Rather, it depends on the class of chemical receptors present on the postsynaptic neuron. In principle, a single neuron releasing a single neurotransmitter can have excitatory effects on some targets, inhibitory effects on others, and modulatory effects on others still. For example, photoreceptor cells in the retina constantly release the neurotransmitter glutamate in the absence of light. So-called off bipolar cells are, like most neurons, excited by the released glutamate. However, neighboring target neurons called on bipolar cells are instead inhibited by glutamate because they lack typical ionotropic glutamate receptors and instead express a class of inhibitory metabotropic glutamate receptors. When light is present, the photoreceptors cease releasing glutamate, which relieves the on bipolar cells from inhibition, activating them. This simultaneously removes the excitation from the off bipolar cells, silencing them. It is possible to identify the type of inhibitory effect a presynaptic neuron will have on a postsynaptic neuron, based on the proteins the presynaptic neuron expresses. Parvalbumin expressing neurons typically dampen the output signal of the postsynaptic neuron in the visual cortex, whereas somatostatin expressing neurons typically block dendritic inputs to the postsynaptic neuron. Neurons have intrinsic electroresponsive properties, like intrinsic transmembrane voltage oscillatory patterns. So neurons can be classified according to their electrophysiological characteristics. Tonic or regular spiking. Some neurons are typically constantly tonically active, typically firing at a constant frequency. Example, interneurons and neurostriatum. Phasic or bursting. Neurons that fire in bursts are called phasic. Fast spiking. Some neurons are notable for their high firing rates. For example, some types of cortical inhibitory interneurons, cells in globus pallidus, retinal ganglion cells. Neurotransmitters are chemical messengers passed from one neuron to another neuron or to a muscle cell or gland cell. Cholinergic neurons, acetylcholine. Acetylcholine is released from presynaptic neurons into the synaptic cleft. It acts as a ligand for both ligand-gated ion channels and metabotropic GPCRs. Muscarinic receptors. Nicotinic receptors are pentameric ligand-gated ion channels composed of alpha and beta subunits that bind nicotine. Ligand binding opens the channel, causing influx of Na+ depolarization and increases the probability of presynaptic neurotransmitter release. Acetylcholine is synthesized from choline and acetyl coenzyme A. Neurons communicate with each other via synapses, where either the axon terminal of one cell contacts another neuron's dendrite, soma, or, less commonly, axon. Neurons such as the Purkinje cells in the cerebellum can have over 1,000 dendritic branches, making connections with tens of thousands of other cells. Other neurons, such as the magnocellular neurons of the supraoptic nucleus, have only one or two dendrites, each of which receives thousands of synapses. Synapses can be excitatory or inhibitory, either increasing or decreasing activity in the target neuron, respectively. Some neurons also communicate via electrical synapses, which are direct electrically conductive junctions between cells. When an action potential reaches the axon terminal, it opens voltage-gated calcium channels, allowing calcium ions to enter the terminal. Calcium causes synaptic vesicles filled with neurotransmitter molecules to fuse with the membrane, releasing their contents into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and activate receptors on the postsynaptic neuron. High cytosolic calcium in the axon terminal triggers mitochondrial calcium uptake, which in turn activates mitochondrial energy metabolism to produce ATP to support continuous neurotransmission. An audapse is a synapse in which a neuron's axon connects to its own dendrites. The human brain has some 8.6 times 10 to the 10th, 86 billion neurons. Each neuron has, on average, 7,000 synaptic connections to other neurons. It has been estimated that the brain of a 3-year-old child has about 10 to the 15th synapses, 1 quadrillion. This number declines with age, stabilizing by adulthood. Estimates vary for an adult ranging from 10 to the 14th to 5 times 10 to the 14th synapses, 100 to 500 trillion. Beyond electrical and chemical signaling, studies suggest neurons in healthy human brains can also communicate through force generated by the enlargement of dendritic spines, the transfer of proteins, transneuronally transported proteins, TNTPs. They can also get modulated by input from the environment and hormones released from other parts of the organism, which could be influenced more or less directly by neurons. This also applies to neurotrophins such as BDNF. The gut microbiome is also connected with the brain. Neurons also communicate with microglia, the brain's main immune cells, via specialized contact sites called somatic junctions. These connections enable microglia to constantly monitor and regulate neural functions and exert neuroprotection when needed. In 1937, John Zachary Young suggested that the squid giant axon could be used to study neuronal electrical properties. It is larger than but similar to human neurons, making it easier to study. By inserting electrodes into the squid giant axons, accurate measurements were made of the membrane potential. The cell membrane of the axon and soma contain voltage-gated ion channels that allow the neuron to generate and propagate an electrical signal and action potential. Some neurons also generate subthreshold membrane potential oscillations. These signals are generated and propagated by charge-carrying ions, including sodium, Na+, potassium, K+, chloride, Cl-, and calcium, Ca2+. Several stimuli can activate a neuron leading to electrical activity, including pressure, stretch, chemical transmitters, and changes of the electric potential across the cell membrane. Stimuli cause specific ion channels within the cell membrane to open, leading to a flow of ions through the cell membrane, changing the membrane potential. Neurons must maintain the specific electrical properties that define their neuron type. Thin neurons and axons require less metabolic expense to produce and carry action potentials, but thicker axons convey impulses more rapidly. To minimize metabolic expense while maintaining rapid conduction, many neurons have insulating sheaths of myelin around their axons. The sheaths are formed by glial cells, oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system. The sheath enables action potentials to travel faster than in unmyelinated axons of the same diameter, whilst using less energy. The myelin sheath in peripheral nerves normally runs along the axon in sections about 1 millimeter long, punctuated by unsheathed nodes of Ranvier, which contain a high density of voltage-gated ion channels. Multiple sclerosis is a neurological disorder that results from demyelination of axons in the central nervous system. Some neurons do not generate action potentials, but instead generate a graded electrical signal, which in turn causes graded neurotransmitter release. Such non-spiking neurons tend to be sensory neurons or interneurons because they cannot carry signals long distances.